Hello students. So in today's session, we will be discussing the topic gingival inflammation. So this is a very small topic, but this is a very important topic too. So it is very important that you understand this topic properly. So I have tried to incorporate more of pictures so that you will understand this well. So now you all have learned in your physiology, right? What is inflammation? Inflammation can be defined as a local response of living mammalian tissues to injury due to any agent. So it is a local response of living mammalian tissues to injury due to an agent. So the inflammation of the gingiva, it is known as the gingivitis. Now, what is the primary etiology for gingivitis? It is the dental plaque. Now, there are other causes too. What are the other causes? It can be caused due to systemic diseases, allergies, medications, hormones, viruses. But the primary cause is because of the dental plaque. So in this chapter, the question that will come to you is, what are the stages of gingival inflammation? Okay. So the sequence of events that occur during the development of gingivitis, it mainly occurs in 4 different stages. The first one is the initial lesion, the second one is the early lesion, the third one is the established lesion, and the fourth one is the advanced lesion. So before coming to the stages, the first stage, see, this is the cross section of the tooth, and this is a tooth. And this is a gingival epithelium. This is the sulcular epithelium, and you can appreciate here, this is the junctional epithelium, okay? And this brown thing which is present here, it is the plaque, okay? So the plaque that was present on the tooth, okay, it will trigger inflammation. So here, now we are coming on to the stage 1, that is the initial lesion. So the plaque that was present on the tooth, it triggers the inflammation. The bacteria and the toxins, they penetrate through the intercellular gaps of the junctional epithelium and they enter the connective tissue. See this picture. This is the bacteria and their toxins. They are entering the connective tissue through the intercellular spaces of the junctional epithelium, and they will stimulate the neutrophils and the mast cells, and there is a release of vasoactive mediators. And what they will do, it will lead to vasodilatation. There is an increase in the blood flow. So the dilatation of the capillaries, arterioles, and the venules is one of the prominent feature in the initial lesion. So you can understand it from this picture itself. Now, the vasoactive mediators, along with the vasodilatation that it causes, there is also an increase in the vascular permeability. See this picture, students. There is an increase in the vascular permeability, okay? And that leads to the escape of plasma fluids containing proteins. See, there is an escape of the plasma proteins, okay? So this is called as the exudate, and that escapes through the sulcus, okay, as the GCF. So there is an escape of the plasma proteins through the sulcus as the GCF, that is gingival crevicular fluid, okay? So now coming on to the next picture. In this picture, we can see that there is margination, adherence of the neutrophils to the vessels, leading to diapedesis, escape of the neutrophils, and chemotaxis. The escape neutrophils, through a process called chemotaxis, migrate to the area of insult. Okay, they are migrating to the area of insult, and in this picture we can see that the neutrophils, they accumulate below the junctional epithelium, and we can also see that they are infiltrating the junctional epithelium. Okay, so this was our stage 1. Okay, so this stage, it shows no visible changes except for the fact that there is an exudation of the fluid from the gingival sulcus. Okay, That's why this stage is also called as the subclinical gingivitis. Now we are coming to the next stage, that is the early lesion. So the early lesion, it starts within 4 to 7 days of plaque accumulation. And which are the most prominent cells? The most prominent cells are the lymphocytes and the polymorphonuclear neutrophils. The lymphocytes are the predominant cells here, okay? 75% of the cells are the lymphocytes. And we can see small amount of very few plasma cells also. Now, this lymphocytes, okay, they release growth factors. So there is angiogenesis, there is proliferation of capillaries, and there is increased formation of capillary loops between the retipex. See, there is a proliferation of capillaries and there is increased formation of the capillary loops between the retipex. Okay, thus bleeding on probing may be one of the important findings which we can see in early lesion. Now, microscopically, there is a dense infiltration of the junctional epithelium by neutrophils. See, here the junctional epithelium is densely infiltrated by neutrophils, and we can see neutrophils engulfing the bacteria in the sulcus. Okay. Now, after the ingestion of the bacteria, the bacteria is broken down by enzymes, okay? However, some of these enzymes, these are also released extracellularly. And what will happen? They will damage the collagen and the blood vessels. So, 70% of the collagen will be destroyed. So, predominantly, it is the dendrogingival fibers and the circular fibers that are destroyed. Then, one another finding which we can see in the early lesion is that this inflammation, it will trigger the formation of reddy ridges in the junctional epithelium. See, we can see in this picture, you can appreciate the formation of reddy ridges in the junctional epithelium. Okay, so that was our stage 2. Now we are coming to the stage 3, which is the established lesion. Okay. So, in established lesion, the extravasation of the red blood cells into the connective tissue takes place, and there is a breakdown of hemoglobin into its component pigments that deepens the color of the gingiva. Okay, so there is an extravasation of the RBCs in the connective tissue, and there is a breakdown of hemoglobin which will deepen the color of the gingiva. And this stage is predominantly, it is dominated by our plasma cell. Clinically, this stage, the gingiva is edematous and it is either deep red or bluish red in color, and the bluish hue is due to the sluggish blood flow. Okay. Then, in established lesion, we can see that there is further proliferation and formation of reddy ridges in the junctional. There is further formation and proliferation of reddy ridges in the junctional epithelium, and there is further destruction of the collagen fibers. Okay. Now coming on to the next stage, the last stage, that is the advanced lesion. So, this is the final stage. And the inflammatory cell infiltrate, extends laterally and further apically into the connective tissue. So, the advanced lesion has all characteristics of established lesion, okay? But it differs importantly in that the alveolar bone loss occurs and the fiber damage is extensive, and the junctional epithelium migrates apically from the cemento-enamel junction. And it is generally accepted that the plasma cells are the dominant cell type in the advanced lesion. Okay, so with this, we come to the end of our chapter, that is gingival inflammation. So these are the 4 stages: initial, early, established, and advanced lesion. So the initial lesion, it occurs, this is a summary, okay, this is a summary. So the initial lesion, it occurs within 2 to 4 days. Then in the blood vessel, early lesion, for the early lesion to get formed, it takes 4 to 7 days, and established lesion, it is 14 to 21 days. And Coming on to the blood vessels, I have told you in the initial lesion, what happens? It is a vascular dilatation that takes place. And in the early lesion, we can see that there is vascular proliferation. And in the established lesion, it is same as stage 2, plus there is blood stasis. Then coming to the cells which predominate, this is very important. Examiners keep asking in your viva. So the cells that predominate in the initial lesion is the neutrophils, whereas in the early lesion it is the lymphocytes, and in established lesion it is the plasma cells. Okay. Then coming on to the clinical findings, we cannot see clinical findings in initial lesion. That's why we call it as subclinical gingivitis. Whereas in the stage 2, we can see bleeding on probing. So that is also an important question which comes in your viva. In which stage we can see bleeding on probing? So it is in stage 2 where we can appreciate bleeding on probing, and in established lesion there is changes in the color, size, texture, etc. So with this we come to the end of our chapter, gingival inflammation. Thank you all.